Hello, and welcome back to part 2 of this series of lectures on scientific ethics, where we're taking a look at alternatives to human organ transplants. And hopefully you're back to find out what xenotransplants are. So xenotransplants are basically transplants or the implantation of non-human animal cells into a human recipient. And so we're basically taking it across different species. And so the idea is that xenotransplants are viewed as a potentially acceptable source of biological tissue and organs when the human sources are limited. And so the idea is that you can go and get an animal organ if you don't have a human organ that's available. Potentially, this is when it's human sources are limited. Also, if there are a lot of ethical concerns associated with the human sources. And so fetal tissues, which we'll talk a little bit with stem cells coming up in the next couple of lectures. And so xenotransplants, again, taking it from a non-human animal and putting it into a human recipient. Now, the problem with the xenotransplantation is this risk of a zoonotic infection. A zoonotic infection, as we've heard a lot about within the last year or so, is an infection or disease that crosses species. And so the risk is that the animal tissue has a pathogen, a disease-causing material in it, that once it gets into a human being is going to cause the disease in the human being, and potentially being kind of a new or unknown pathogen can then spread, kind of similar to what we see with COVID-19. This is a zoonotic infection. COVID-19 is a zoonotic infection that was originally in bats and has now made the jump into human beings and is causing great disruption and concern kind of globally. And so again, there's this fear that if we're transplanting between an animal and a human being, we could be potentially causing kind of a new disease to become established within humans. There's also a risk of potential tumor formation as these foreign cells, these animal cells, become established within the body. This tumor formation is bad. It's bad for the recipient, but the zoonotic infections is bad for the patient as well as society. As we can see, if you have this zoonotic infection, it could be potentially very infectious, contagious, and then spread throughout society. And so because of that, patients that receive these xenotransplants, these transplants of animal tissues, often are going to be subject to special conditions. And so recipients of a xenotransplant are prohibited from doing blood donation. Again, there's this fear that something within these animal cells then could be spread within the bloodstream to other individuals. And this actually extends to close contacts of those that have received xenotransplant. And so, a close family member that has a xenotransplant would prevent you from becoming a blood donor because there's a fear that you would be exposed to something by the patient that got the animal tissues, and you could be, a carrier for this potentially zoonotic or disease that's spread from the animal to the human. Patients that also receive xenotransplants have to agree to lifelong monitoring. They have to, come back for kind of checks to make sure that they don't have a disease process that's being established within their body that could potentially, spread throughout society. So examples of xenotransplantation, again, many of these are, before some of the modern advances of medicine. But we saw in the 1960s, chimpanzees' kidneys going into patients in kidney failure. Baboon hearts were transplanted in the 80s from a baboon into a human being. Related to that, a baboon liver was transplanted into patients in liver failure. And then in the 1990s, we were taking a look at pig tissue transplants. We saw pigskin being used as a source for burn patients. as well as this idea that we could get pig neuronal cells, and this is getting into some of those ethical issues that I talked about, like the stem cells, which we'll come back to in a couple of days, taking some of these pig neuronal cells for treatments of Parkinson's disease and Huntington's disease. Both of these are examples where we have loss of cells within the nervous system. We want to try to reestablish those cells where it's not normally possible to get kind of human brain cells and inject them in. Now, in most cases, pigs are the preferred source for xenotransplant tissues and organs. Pigs are relatively quick to mature. They're bred for captivity. We've had lots of extensive interactions between humans and pigs for a long period of time. Pigs have been a pretty reliable food source throughout human history. So the idea, because we've had this close interaction with them, it's less likely that there's a disease that these pigs could be carrying that potentially could then spread to a human being. We also take a look at it. Pigs are comparable. Their organs, the organs we're going to be interested in transplanting, their hearts, their livers, things like that, are going to be relatively the same size as a human being. And so therefore, we got a good size match going in. Still, like all of the other transplants we've talked about, we've got immune system concerns. Pig cells are going to be very, very different. They're going to have very different molecules, very different proteins on their surface. Think about the blood cells where we had the purple popsicles and the bluish green squares. They are going to have lots of little molecules on their cells that could be recognized as being foreign in the recipient. And so again, the xenotransplants have the same concerns as a poorly matched human-to-human transplant. So we want to make sure that the donor is healthy. In this case, we're looking at potentially a pig, but you want to test and make sure that the pig doesn't have a virus or either infectious diseases or anything going wrong with it that would then be injected into and causing disease within the human recipient. And you're also concerned, very, very rightly so, on immune system rejection. We're injecting in, we're putting in, transplanting in foreign tissues. And they're going to be recognized as foreign, but we're going to be doing this because we don't have a better alternative. So that means then is that patients that get these xenotransplants are going to be on immunosuppressive drugs. They're going to have their immune systems kind of turned down, so they're going to be very susceptible to catching illnesses, catching diseases of other types, especially infectious diseases. But again, keep in mind, we're doing a xenotransplant because they're probably going to die if they don't get an organ at this period in time. Now, if we take a look at it, pig to human transplants are often associated with either hyperacute or delayed rejection. In most cases, these xenotransplants are going to be a temporary stopgap until we can get a human organ that's going to be present. Because the human immune system, as we've talked about before, when we think about the purple popsicles and the blue-green squares, they're going to recognize these foreign molecules, these foreign shapes on these cells. And even with immunosuppression, the human immune system is gradually going to break this down. And so we've got an example here of some of the immune system kind of attacking some of these. This looks like a kidney, attacking and killing off some of these cells. Now, one way to treat this is an effort to create strains of pigs that are less immunogenic, with the idea that we're going to be altering these pigs in a way, in some cases altering them genetically, so that they're going to have less of these purple popsicles and blue-green squares on them. So less of these immune system triggering molecules along their surface, but basically, make them so that they're going to be generating these organs that are more human-like. So almost like a little chimera, a little human organ farm that's being created within the pigs they're growing. Again, potential ethical issues associated with that. Are we making these pigs kind of like subhuman if we're changing them in a way that they're going to start expressing human molecules on their surface as well? Readings associated with this topic, very nice Guardian articles, using animal organs in human. It's just a question of when. This is something that's going on, has been going on. We're working at trying to come up with better ways of doing it. The xenotransplantation ethics debate, using animal organs for humans. And then finally, a Vatican report, Prospects for Xenotransplantation, Scientific Aspects and Ethical Considerations. So again, a little bit extensive on the readings, but I think this is going to be important for you to take a look at. Not too bad. They're designed for a broad audience. The Vatican one's a little bit long, but read what you can, especially the first two. I think are going to give you a good flavor for what it is that we're talking about with xenotransplantation. As always, if you have any questions, feel free to email me. Thank you and have a great day.